Everyone, and welcome to our online learning. We're going to get into genetics and talking about how traits are passed on from generation to generation. So what you guys should have done is you should have downloaded the notes or open up a document onto your screen so that you can type the notes as we go through. At a certain point along the way, I have a mini YouTube video that I'll share with you that I think animates the process pretty nicely. And by the end, we'll be doing some example genetics problems. Now, I know that you guys did some problem solving last week as well, but I thought it would be a good idea for us to do some together in a more formalized way and to review. So without any further intro, let's just go ahead and get started. So with genetics, branch of biology that studies the way hereditary information is passed from one generation to the next, it really all starts out with this man named Gregor Mendel. And Gregor Mendel did these studies. Here he is. Gregor Mendel did these studies with pea plants. So, trying to move it so that you guys could still see all the text. So, Gregor Mendel was an Austrian monk, and in the garden at his monastery, he did a number of genetic crosses involving pea plants. So, pea plants were pretty ideal for genetic studies for a number of reasons. Even though pea plants are pretty simple, and our natural interest is probably in ourselves and studying human traits, pea plants would really lay the foundation for our understanding of how traits are inherited and passed on from one generation to the next. In his time, people were able to recognize how offspring are a blend of their parents, but in reality, we know that DNA carries genes, and then those units are passed on to offspring, and we have one allele from mom and one allele from dad at each gene. So back to the plants made themselves uniquely easy models of study because they're easy to grow, they mature very quickly, they are easy to pollinate, easy to reproduce, and they have distinct characteristics. So there are certain models in biology that present themselves as ideal models to study because it is easy to get a large body of quantitative data from which to draw conclusions. So in humans, we don't grow and mature quickly. It would not be easy to control reproduction. We have a wide variety of characteristics. Our skin color is not just dark or light. We don't just have one or two shades of hair colors, but pea plants are easier to study. Some of the traits that Mendel studied includes the shape of seeds. Are they round or wrinkled? The color of seeds, yellow or green. The color of pods, green or yellow. All the way through to flower color. Is it purple or is it white? And everything you see in this top row here, these are all the dominant traits. And everything down here, these are all the recessive traits. So we can make some generalizations from Mendel's studies and apply to some human traits as well. Now, there are some important terms if we are going to better understand genetics. But before we get that, unlike other scientists, Mendel used numbers and ratios in order to understand his data. So by performing all these experiments and getting and doing so many trials with many offspring, he was able to obtain large numbers and draw conclusions. You see this number F, this term F2. You see this F1. What does that mean? After I show you the TED-Ed video, I think it'll make a little bit more sense. You guys will be seeing this TED-Ed video, and you'll be seeing some of these terms in that video. So I just have to move myself here. So important terms that you'll see as we get further into genetics is the term allele. So an allele is just a version or form of a gene that controls a trait. These are a homologous pair of chromosomes. This and this, both of these, are single chromosomes. And here is the locus for the flower color gene. A locus is just a very fancy term for just the gene location. So on this chromosome is the allele for purple flowers, and on this chromosome is the allele for white flowers. This may have come from the maternal parent. This could have come from the paternal parent, or vice versa. Nevertheless, this is the gene for flower color. And at this gene is the allele for purple flowers, and at this gene it's the allele for white flowers. And in this example here, for height, big T is the allele for tall, and lowercase t is the allele for short. So there can be dominant alleles and there can be recessive alleles. So a dominant allele will show up and express its presence no matter what other allele is present. A recessive allele will only express itself if there's two recessive alleles present. So big T, a capital T, that would be dominant, and we represent the alleles capital for dominant. And in this case, that could be tall. Lowercase t, in this case, that would be short, and that would be the recessive trait. As you guys may have noticed in some of the example problems that you did, these other terms, genotype and phenotype. So in order to remember what these terms mean, within the term genotype, you see the first three letters, G-E-N, so that refers to the genes that the organism has. It's the actual genetic representation of a trait, the two alleles that that organism has. The phenotype is the physical appearance, the physical manifestation of that trait. And you can recall that, or you can remember that because the pH in phenotype, you can think of the pH in physical. So over here in this picture, we see that this P that has two capital S's for smooth, it is a smooth P. It has the dominant allele. This P here has just one of the dominant alleles for smooth, and it still has the same phenotype. So both of these P's have the same phenotype despite the fact that they have different genotypes, because both of these P's still have the dominant S allele for smooth. In this case here, this P does not have any dominant S allele for smooth. It just has two of the recessive S alleles for wrinkled. And the phenotype that corresponds to this would be a wrinkled appearance. So just because it is showing the dominant phenotype doesn't necessarily mean that we could determine the genotype. There's one of possible two genotypes here. Over on the bottom here, we've talked about blue and brown eyes numerous times. And here the phenotype is blue eyes. Here the phenotype is brown eyes. So again, phenotype is the observable trait. Now, you'll see that there are these other terms called homozygous and heterozygous. Homozygous is when there's two of the same allele, and heterozygous is when there are two different alleles. Now, on this left-hand side in this picture, these are all the phenotypes. These are all the physical appearance. On the right-hand side here are all of the different genotypes. So these three flowers are clearly purple, and then this flower is clearly white. On the right-hand side, when we refer to genotype, Each one of these purple flowers, in order to be purple in the first place, must have the dominant P allele, because in this case, purple is dominant over white. So it has the dominant P. And then in this bottom situation, this flower is white because it doesn't have the dominant P allele. Now to tie in the new terms of homozygous and heterozygous. These purple flowers may have one or two of the dominant P allele. In this first flower over here, it has two dominant alleles, so two of the same. And so we say it's homozygous because homo means same. So big P, or uppercase P and uppercase P. For these two flowers here, these flowers both are what we call heterozygous. They're still purple, just like this one is, but they do not have two of the same alleles. Each one of these has one of the uppercase P and one of the lowercase p, which is one of the purple allele and one of the white allele, making them heterozygous. So, going to Mendel's experiments, I think the best way to show this to you is through this TED-Ed video. It does a very good job of explaining Mendel's pea plants in probably a pretty cute way. So without any further ado, I'll just show that to you. And here.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded.
So he bred the yellow and the green pea, and he might have expected either that there would be a mix of yellow and green, or that there would be a set of offspring that are yellowish green in color. However, to his surprise, this is what he got.
Plant, and he got only yellow seeds.
So the question is, where did that green trait go? Why are there only yellow peas in that generation?
He called the yellow color trait the dominant one, because it was expressed in all the new seeds.
So this would be Mendel's law of dominance. This is Mendel's law of dominance because the yellow trait would appear over the green. It seems like the green trait is therefore hidden.
Then he let the new yellow-seeded hybrid plants self-fertilize.
So one would expect that if we have these two yellow peas, that all of the offspring would be yellow. However, to Mendel's surprise, he did not get all yellow offspring.
And in this second generation, he got both yellow and green seeds.
So then, in this case, it seems that the recessive trait reappeared.
Which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele.
So in this Punnett square, it's basically looking for the probability that different outcomes would be predicted from this genetic cross. So these two yellow peas are both heterozygous. This P was big Y, little y, and each column here has one of those alleles.
As an uppercase y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous P mom will give each P kid a yellow dominant allele, and the green homozygous P dad will give a green recessive allele, so all the P kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited. But it all started with Mendel and his peas.
So I thought that this video did a pretty good job of explaining how that all worked. And here is a similar experiment to what Mendel did, just with a different trait. So in the TED Eds that you guys saw, it was just with yellow and green peas. And here it is just with purple and white flowers instead, but the idea is still the same. So this starting generation is the P generation, and here P stands for parent. So the parents are purple flowers and white flowers, and they are both homozygous. So this would be a capital P for purple and a capital P for purple. This would be a lowercase p for white and a lowercase p for white. So they're both purebred. So if Mendel crossed this purple flower with this white flower, he might have predicted that in the next generation that they would be purple and white, or maybe a light purple color. However, in this, what we call F1 generation, which stands for first filial generation, he observed that all of the flowers, all of the plants had purple flowers, indicating that that white trait was now hidden in this generation. Then what Mendel did was he took two of these purple flowers, he crossed them together, and he might have expected that they were all purple in the next generation. But in this next generation, what he observed, which we call the second filial generation, hence F2, he noticed that the white trait reappeared. So he got mostly purple flowers, because that's the dominant trait, but he noticed that the white flowers reappeared. So these are Mendel's experiments. And Mendel determined his laws. So one law is the law of dominance, and that is that some alleles are dominant and cover the recessive alleles. One trait masks the effects of the others. So in the TED video that you guys saw, the yellow trait shows over the green trait. In the previous slides that I showed, the purple flowers, which the purple allele would show over the white allele. The other law that Mendel concluded was the law of segregation. It's the idea that when gametes are formed, the alleles for each gene separate, so that each gamete receives only one allele from each pair. It's something that you guys already have envisioned when we were learning about meiosis, but this is putting a formal term to it. So for example, a parent who is heterozygous, so a capital Y and lowercase y, creates one gamete with a dominant allele for yellow and one gamete with a recessive Y allele for green. And that's what this picture shows. So the parent who's heterozygous produces gametes that have both the dominant allele and the recessive allele. The third of Mendel's laws is the law of independent assortment, and we explored that as well when we were learning about meiosis. This law states that alleles for different traits are distributed to sex cells independently from one another. So in other words, simply stated, alleles on different chromosomes are not linked. So to sum it up simply, genes are not linked. We know today that they're linked, but this is something that Mendel had concluded. So this is the takeaway idea. Just because the seed is yellow does not mean that it will always be round. And just because the seed is green does not mean it will always be wrinkled. So here's an example with a pea that's both round and yellow. And here, a pea that's green and wrinkled. So the next generation may have peas that are all hybrids, but if we bred the two hybrids together, in the following generation, we'll get a whole assortment of different traits. So here's a better illustration of the law of independent assortment. I think that I really liked this picture best because it shows each of the principles. So here in green for segregation, it shows how the chromosomes during gamete formation will separate from each other. In blue, independent assortment, the idea is that the chromosomes are not linked to each other and they can shuffle into the different gametes in a variety of different combinations. And then in purple here, the principle of dominance relates to the idea that the dominant allele masks the effect of the other allele, which is the recessive. So here, purple is dominant over white, and tall is dominant over short. Now, importantly, is the idea of problem solving. So here are some examples. So steps for problem solving: underline the phenotypes mentioned in the problem, step 1. Step 2, based upon the problem, write down the genotypes that would be crossed. For example, here we have heterozygous round and heterozygous round. Number 3, set up the Punnett square with one parent's genotype on each side, and then answer the relevant questions. So here we set up the heterozygous round, capital R and lowercase r, here. And then here we filled out the Punnett square. Then, in order to determine the genotypic and phenotypic ratios, which really are just basically probability ratios, you determine the number of each type. So here we have one homozygous dominant, so one big R, big R. We have 2 of the heterozygotes. We have one homozygous recessive. So the probability ratio for genotypes is 1 to 2 to 1. Here we have 1, 2, 3 possibilities where round is dominant and 1 possibility where wrinkle would be the phenotype. So the probability ratio for phenotype is 3 to 1. And then overall, the probability that we get something that's round is 75%. The probability that we get wrinkled is 25%. So now let's do another problem. Let's do another example. So brown eyes are dominant to blue eyes. So here I'll just put brown eyes is dominant over blue. So here's brown and here's blue. Mom is heterozygous and dad has blue eyes. So if mom is heterozygous, then that means that she would be capital B lowercase b, and then dad has blue eyes, so he would be homozygous recessive. So these are the genotypes for the parental cross. The question is, what is the probability that their offspring will have blue eyes like dad? So eye color is inherited in a bit more of a complex way because eye color is polygenic, but we'll go with this as an example. So in order to set up the Punnett square, here is our 4-box square, and we just put one allele on top of each column, and then one allele next to each row for the other parent, and just fill in. So here we have capital B, lowercase b, capital B, lowercase b, lowercase b, lowercase b, lowercase b, lowercase b. Now what we need to do is answer the question. So the question asks, what is the probability that their offspring will have blue eyes like dad? So this would be brown eyes. This would be brown eyes. So brown eyes is the phenotype that goes with a genotype, capital B, lowercase b. Brown eyes is the phenotype that goes with the genotype, capital B, lowercase b. Here, this is blue, this is blue. So the answer to the problem here is 50%. So the answer here is 50%. Now, we need to determine the genotypic ratio. So there's two possible genotypes that we obtained here. So we have, as possibilities, a heterozygote and a homozygous recessive. So we have 1, 2 heterozygotes, which are capital B, lowercase b, and 2 homozygous recessives, lowercase b, lowercase b. You could leave the genotypic ratio as is, which is 2 to 2, or you may reduce it further, which is 1 to 1. Either or is acceptable, but ultimately, okay, the answer is 1 to 1. Then the phenotypic ratio here, there's 2 phenotypes that we have. One phenotype that we have is brown, and we generally put the dominant phenotype first. The other one that we have is blue. So there's 2 possibilities where the outcome is brown, and there's 2 possibilities where the outcome is blue. So our phenotypic ratio is 2 to 2. Again, you can reduce this to 1 to 1, although 2 to 2 is not incorrect either. And the next problem is with hitchhiker's thumb. So on the left side here, that's what hitchhiker's thumb looks like, and it is a dominant trait, and a straight thumb is recessive. So hitchhiker's thumb is dominant to a straight thumb, cross a homozygous dominant genotype. So you could use, why don't we just use T? So homozygous dominant genotype with a heterozygous genotype. So we're crossing these 2 possibilities. What are the genotypic and phenotypic ratios? So here's the parents for our cross, and here is our Punnett square. So we have homozygous dominant with 2 dominant alleles. We have the heterozygote. Now we can fill in the square as so, and now answer the question. So the genotypic ratio, we have 2 genotypes that we determined which were possible, homozygous dominant and heterozygous. So we see that there's 2 homozygous dominants, and we see that there's 2 heterozygotes. Again, there's a 2 to 2 ratio. You can reduce that to 1 to 1. Then, for the phenotypic ratio, the 2 possible phenotypes are hitchhiker's thumb to a straight thumb. Now, in this situation, each one of these is a hitchhiker's thumb. So we have 4 hitchhiker's thumbs in our Punnett square and none of the straight. So actually in this situation, we have a 4 to 0 ratio. It's possible that you could reduce it to 1 to 0. Now, some of you may bring up in math how you may not, you would reduce it differently because the 0 is on the right side of the ratio. In terms of genetics for our ratios, It's okay in genetics to do a 4-0 or 1-0 ratio. If you have questions about that, email me if I did not make that clear. Now, it's important that we go through some examples, and I'm not going to, I wanted to just go through some examples with you guys because I need to do that in order to make clear the vocabulary. So, okay. So, for example, for the seed coat shape trait. So here's the seed coat shape trait. What is the genotype of a homozygous dominant organism? So if it's homozygous dominant, homo means that it's the same of the dominant allele. So we're looking at the letter big R, little r. So that would be capital R, capital R. For 1b, the phenotype of a homozygous dominant organism, okay, that would still be big R, big R. I should have changed that to heterozygous. For 1c, the genotype of a homozygous recessive organism. So if it's homozygous recessive, it would have 2 of the recessive R's. For 1d, what would be the phenotype of a homozygous recessive organism? So if it's homozygous recessive for seed coat shape, here in this case, it would be wrinkled. What would be the genotype of a heterozygote? So the genotype of a heterozygote would be big R, little r. The phenotype of a heterozygote would be, in this case, round. And I'll just go through a couple of these problems, not every single one, but just to give you a general idea of how these are done. So for this one, number 3, here we are crossing big T little t with big T little t. Now, these are 2 heterozygotes that are being crossed. So when crossing these 2, it's the same way we set it up as when I showed you guys in the PowerPoints. So here we have big T, little t, and still fill out the square. So here we have something that's homozygous dominant. Here something that's heterozygous. Here heterozygous, and here homozygous recessive. Now let's say that the recessive, the lowercase t is for, capital T is for tall. Let's say the lowercase t is for the short phenotype. So for this problem, now let's determine the genotype and phenotype ratio. So for the genotype, there's 3 that were possible. We see that we could have gotten the big T, we could have gotten the heterozygote, and we could have gotten the homozygous recessive. So we have 1 homozygous dominant, we have 2 heterozygotes, and we have 1 homozygous recessive. So that would be our probability ratio for the genotype. Our probability ratio for the phenotype, well, there's 2 phenotypes possible. So the 2 possibilities are tall and short. So here, anything with a capital letter is tall. So there's 3 possible talls, but there's only 1 possible short. Okay? Now, if we take this, I'm just going to get rid of that. And I want to go to the problem where you're not necessarily given the genotypes immediately, but it's more so of a word problem. So let's say here that we're told that we are given a homozygous tall plant with a short plant. So we're given, again, the words, and we have to determine the genotype. So if it's homozygous tall, we have big T, and if it's a short plant, it didn't tell us if it was homozygous or heterozygous, but because it's short, it has to be the recessive genotype. So it's little t. Now it's business as usual. So set up the Punnett square here, and this is big T, lowercase t. If, as you guys were working, you switch this and this was lowercase t, and that's capital T, that's fine as well. But every single square or little square in this Punnett square has the same outcome. So for the genotype ratio here, we see that they're all the same. So if they're all big T, little t, we see that the ratio is 4:0 because it's like we have all big T, little t and nothing of anything else. For the phenotype ratio, all of them are tall. And it's like we say we have 4 of tall and nothing of anything else. So later I'm going to have you guys do some practice problem solving, and we'll stop with that.